as we go through exam 3, that replication and cell division are tied together. And things that affect replication also affect cell division. But to start out here, we're talking about not replication, but the two other really important molecular processes, transcription and translation. We don't necessarily care about all the details of how these things work. We do need to have a fundamental appreciation for the molecules that are involved and the flow of information. What that means is we start with DNA. And from DNA, we make an RNA molecule, which is what the transcription process does. And then using that RNA, we make a protein. And we call this process translation. Together, transcription and translation accomplish what we call gene expression. And so, particular DNA sequences are transcribed, what we call the gene, and the RNA that we actually make is the messenger RNA. And so there's a little bit more detail as we sort of go along about just remembering some of the connections between things. But taking the information contained within DNA and turning it into a functional actor, the protein, is what gene expression means. And one of the things that we're going to talk more about as we go forward is that this is a highly regulated and controlled process. When you make certain proteins, depends on environmental factors, developmental cycles, all sort of things control when things happen and what cells do what things. And just as an example, take the human body. Do liver cells do the same thing that lung cells do? Do heart cells do the same thing that skin cells do? Every cell in your body has the same DNA. They do different things with it. And that's the important thing about regulation and control. It allows cells to specialize, but we need to control when things happen at various times. When it comes to talking about transcription and translation, don't use the phrase turned into. DNA is not turned into RNA. It's either copied or used, because DNA is not altered or changed during the transcription process. Also, the RNA is not used, consumed, so we don't say the RNA is turned into a protein. We want to say that the RNA is used to make a protein. And we'll already get into some other little things as we go along. But as we think about the messenger RNA, the most important concept in the messenger RNA is the idea of a codon, which is 3 nucleotides. And you get a codon, 3 nucleotides, the next 3 nucleotides are the next codon, the next 3 are the next codon, and so on. And each codon is 1 amino acid. And so there is a specific relationship between the 3 nucleotides in the mRNA that come from the gene and the amino acid that gets put into that certain position. This is where mutations are going to play an important role. If we change the DNA sequence, the RNA sequence gets changed. What effect does it have on the protein sequence? The amino acids that are used to make that final product. So the mRNA contains what we call a codon. The tRNA, which is the delivery device for the translation process, will base pair with the mRNA to specify what amino acid comes next. And it uses what's called an anticodon. This is a base pairing idea we need to think about. Codon and anticodon base pair together, like the 2 strands of a DNA molecule base pair together. They do it anti-parallel and complementary. So the specific interaction of one tRNA with one codon is what puts the right amino acid in the right position of a protein chain. When we think about codons, there are 2 basic types, what we call sense codons and nonsense codons. Nonsense are always what we call stop codons. And there's no tRNA for a stop codon, but there are TNAs for all the other codons, which we call sense codons. One of the most important sense codons is the start codon. And while we're not going to spend a whole lot of time trying to think about the beginning and end of the translation process or the beginning and end of the transcription process, we've already sort of done that for exam 1. You need to understand that certain codons in the mRNA, few amino acids, another one do not. And the nonsense codons. To go from codon to amino acid, we use something called the genetic code. I know some of you have probably heard too much about the genetic code in other classes. But we do need to make sure we all have a certain foundation before going forward. The most important idea of the genetic code that we have right now that will go forward and continue to be really important is the idea of redundancy. It's not true for every amino acid, but almost all amino acids have more than one codon that will specify that particular amino acid. And that's what redundancy means. There's another codon, more than one codon for an amino acid. It's not 100%. It is a fairly high percentage of the amino acids are coded by multiple amino acids. The universal aspect won't really matter for us in this class, but that basically means that every living organism uses the same translation method. And that allows us to take human proteins and express them in bacteria, like to make insulin, or human growth factor, or other important enzymes and proteins that are needed for human health. You could also take a bacterial protein or gene and put it in humans for some reason if you want to. I really need to look at this reference up so I have a picture of it to show you to prove that it's true. But there was an article a while ago, like more than 5 years ago, maybe 10 years ago. But somebody genetically engineered cows, but they inserted the silk production gene from spiders into cows so that the cows will secrete silk in their milk. You can harvest the silk and use it for things. And I think specifically they were using it to make bulletproof vests. For whatever reason, silk's pretty good at bulletproof vests. Imagine that, you can turn a cow into not just a milk beef factory, but a silk factory. Imagine what we can turn you into. We are genetically engineered pigs to grow human organs. We can use those in transplant. So also the fun things, if you want to be a mad scientist, you can be a mad scientist, but still be an actual scientist. It's cool. Hopefully you will hear the sarcasm in my voice. Make sure you memorize all of the codons in the genetic code, especially the ones that are not red and not green. And if you're a fan of the Georgia Bulldogs, like Dr. Greenwood and Dr. Rubal, UGA is in red. It's not codon. But look at this. In other classes, you will need to memorize what amino acid goes with what little one-letter designation, which we don't have to worry about that. If you look at this list, hopefully you see that these all have the same letter, so these all work for the same amino acid. This is called leucine. And then there's two up here that also code for leucine. Here's 6 or 4 for serine, then there's 2 more for serine over here, so there's 6. 4 for arginine, and 2 more here, 6 for arginine. 1 for this one, tryptophan. 2 and 2, and some of them have 3, and some of them only have 1, and that's what we need for redundancy, is having multiple copies. And so for almost every single thing, there's 2 that are the exceptions, tryptophan and methionine. Everything else has at least 2 codons for that single amino acid. And by looking at the genetic code, we can see a trend that'll be really important going forward. And to see this trend, we need to look at the amino acids that only have 2 codons. So let's take a look at this group right here, because there's 2 right here. Between the 2 serine codons, what is different? Yeah, the last nucleotide, a U versus a C. For the same thing is true for arginine, the last nucleotide is different, an A versus a G. What is special and unique about that AG? There was a question on exam 2, and a question on exam 1, and it'll be a question on exam 3. They're both purines, which means they're both purines. We're going to add something to this as we go forward, but they're both purines. Or they're pyrimidines. And so as we look at things of groups of 2, it's always that same pattern. Purine versus purine, pyrimidine versus pyrimidine. I mean, it sort of falls away right here, but that's the only sort of exception to this rule. But that peering to peering, riveting to riveting is an important factor in a lot of things. We'll get into why that is as we go a little bit forward. Here in a second, I'm going to talk about 3 things that are going to be really important. You going to wait? Or you can't wait? Let's vote. Who can wait? Who does not want to wait? It's a trick question. Because you have to figure out what am I talking about, are you waiting for me to tell you the answers and go forward? Or are you waiting because you're waiting in your season, waiting? Because you're waiting.
Waiting for next comments.
If you can wait, does that mean that you want to wait until Friday
Yep.
to get this information? Or you're not waiting because you want to hear it now? Now that I've explained it in minute detail, hopefully you know what the right answer is.
Wait until Friday.
We're gonna wait. Because we can't wait. But if you have questions about your test, these final minutes of class, which are now 11 minutes of extra time, are here for you to talk to me about your test.